Hello, my name is Eric Estasio, and I am presenting this paper that was proximity labeling and SILAC-based proteomic approach identifies proteins at the interface of homotypic and heterotypic cancer cell interactions. The main things here are to try and identify intercell interactions in breast cancer. Some of this work had already been done, but most of it had been done in a 2D environment, not a three-dimensional environment. So the goal would be, how does the cell actually interact fully in the body versus when it can only interact on a one-dimensional plane with other cells in a culture next to it. The main form of analysis that we used was SILAC in conjunction with a few other forms of analysis, but for the purposes of this paper, I selected it because of, one, the breast cancer aspect, my mom having had breast cancer a few years ago, and then for this class specifically because of its connection with SILAC. The procedure, the first steps they did were they cloned key mice cells with the breast cancer types and inserted HRP into them so that they could actually test how it would interact and use it as the sort of baseline protein interaction that they were looking for. Once they had cloned and created their samples, then they cultured them for longer, 48-hour and 72-hour increments to get multiple cultures that were viable, and then they labeled them using a variety of different labels. The most common label they used was heavy amino acids, as those would be easier to detect. Then they created spheroids for analysis through filtering out all of the other gels and liquids that they had used to prepare their cultures and the remnants of the cloning, and added calcium chloride to create a more gel-based structure that would be easier to move through the equipment. Once they had done all of this and prepared all of the samples, they took another step where they took 3 groups of cells and established a monoculture, a homotypic co-culture, and a heterotypic co-culture, each in their own dishes, so that they could get 3 different levels of interaction. From here, they did a mass spectrometry process where the orb trap in data-dependent acquisition mode was used. And then they ran it as a comparison against the human proteome that they had already known, so that they could get a comparison between which proteins in the cloned cells that they had cultured were actually working as compared to those that were in the already known human protein structure. Then we move to their results. First thing they noticed was that both forms of the proteins biotinylated, but the bait cells were completely biotinylated on the surface. They had a higher uptake of the HRP that allowed them to completely biotinylate, as opposed to the prey cells in their cultures that did not fully biotinylate. What they did as a follow-up was that in the presence of BP, they could fully biotinylate prey cells by putting in the bait cells that had already been biotinylated. Then, with BP and hydrogen peroxide were both used, they could identify complete outer proteome used in cell-to-cell interactions, which would allow them to identify both areas of commonality and difference, and what proteins were actually active in the transfer of data and movement of cancer cells. So The main thing they found were in a homotypic cell, they had a much higher MCF7, which was um, the bait cell in most of these, where it had 105 interactions that were entirely unique to it, and the MDA MB231 only had 68. But in the middle, they had 45 interactions that were common between both types of cells. Um, the research, their discussion of it showed that they had interest in all three aspects, but some of the highest was in that center 45, where if there were not significant mutations to the cancer cells, those 45 interactions would be the most useful for creating generic um, alternate proteins and enzymes that could interact with these cells. When we look at heterotypic, the MCF7 is basically just the MDA MB231 in terms of how it interacts, with the vast majority of the interaction between MCF7 being the same as the heterotypic MDA, but the MDA having 173 new interactions that it um, that MCF7 did not have. Importantly, with the heterotypic, something interesting to note is the MCF7 actually loses its interactivity. It only has 60 different proteins that are interacting compared to 150 in a homotypic cell. However, the MDA goes from 113 different interactions to 226 different interactions. It doubles its ability to interact in a heterotypic cell. Then they found a few different key signaling pathways that interact with how the cells uh, interact with different matrices, how they interact with each other, how they transfer data. So the key ones they found were the integrin-mediated pathway, the ERK1 and ERK2 cascades, the semaphorin plexin, the ephrin receptor, and the Rho protein signal transduction. Together, these give a good idea of how these cells actually interact with each other and then with the cells around them. Another interaction that they found was the GPTase, and they found that that one in homotypic was exceptionally high and gave a pretty good line for how those cells, homotypic cells, use GPTase connections to actually move and attack non-cancerous cells. So overall, the net result was that they were able to categorize the complete active sites of two different breast cancer cells in two different homotypic and heterotypic environments and create a model of how these cells actually interact with each other and with the cells around them. Here is the link to the article and all of the authors who contributed to it. Thank you for watching my presentation.